this gives you an idea of the kind of images that are coming out of Spatial Molecular Imager. I think in one talk I called this thing the Hubble telescope of spatial biology, and sort of caught on in some respect. Each one of those little dots you're seeing, we're looking at a melanoma tissue here. Each one of those little dots is an individually counted single RNA molecule in an FFPE tissue section. We're doing about 1,013 different RNA classes at a time here. When I'm looking at this thing, when we launch this next year, you'll be capable of doing about 500,000 cells like this, generating about 1 billion transcripts off a single tissue slice. So that's the scale we're working at. 500,000 cells, 1 billion transcripts at a time. Okay, when we're developing this, actually, it started as a different kind of project. It started as a project where I was inventing and developing ultra small cyclic optical barcodes that do sequencing. And it turned out we came up and we developed a 20 nanometer sized a barcode, optical barcode that will change colors as I put different hybridization reagents on there. And it's 4 different colors. So you encode information like that, 4 colors, and then how many bits of information is how many cycle events you want to have, and all the light dark patterns. We developed that early in 2016, and we were going to be developing a clinical gauge sequencer with that. We actually published on some infectious disease detection with that, and in Nature Medicine. I think that was a paper we did, forget what year that was, 2019, I believe. But as we're doing that, I was thinking it's a total hybridization-based sequencing reagent. And then if I look at what I'm doing with that, this is sort of what I was doing with that reagent originally. Look at the left-hand side of your screen here. You know, I was taking Mother Nature's FFPE, yanking the RNA out of it, putting it down onto an ordered array, and then using a hybridization sequencing reagent and sequencing all these things in parallel. And you really think about it, it's like, why go to the work of pulling it out of the tissue? Wouldn't you rather just take Mother Nature's ordered array, which is the tissue itself, leave the RNA where it is, and let's bring that hybridization unit into the tissue. And then now we're quantitating the RNA and High-plex. Now we know where it is. So that's what we did. Starting about 2.5 years ago or so, 3 years ago, we started adapting this to do in situ detection of RNA. And I'll show you how it works now. It works just like all the other nanostring technologies. We built all these chemistries to work on FFPE. It's always the hardest one. My figure is just do the chemistry right, do the hardest one, and the rest of them fall out. So that's what we did here. It's the same thing we did with Genomics. So you just take a standard FFPE slide, you do a standard prep on it. You could even do it on an automated like a bond tissue processor if you want, through the in situ hybridization of these reagents. And I'll show you how the little in situ tagging thing works. Goes into a flow cell, put it into the instrument, you close it up. Actually, it's what the outside of the spatial molecular imager looks like. There's a kit that goes in, you run your reagents, you push your button, and then what you get out are these things to the right. So the information content, obviously all your tissues, you got your 1,000-plex RNAs, so you can do your UMAP clustering. All the UMAP cluster all have X, Y, and Z coordinates to them. So you take all your clusters and you put them back into space. That's what the actual raw data looks like. And then you can do a whole series of different types of analyses at the single cell level, and I'll show you what some of that looks like. But it's a single tabletop box that gets you from A to Z here. I'm going to bring you under the hood on how the chemistry works a little bit. So kind of be able to, it's fun to be able to talk to a technology group like this. So anyway, so here goes. If you know how Genomics works, you got the hybridization, in situ hybridization. Then there's a little tail that's got the sequencing barcode on it, connected with a photocleavable linker. So we do our hybridization, and then we put the light on, and a little tail comes off, and then we sequence ex situ, if you will. But on here, we wanted to bring the imaging reagents in. So we do our hybridization. There's a little tail on what we're hybridizing on. You can sort of see it over here on the far left. So there's Mother Nature in red, the little RNA there. Here's our business end hybridization. There's a tail that sticks off of that hybridization complex. And what I'm going to have, my sequencing barcodes, that little 20 nanometer little sequencing barcode rides on the back of a hybridization reagent that goes cycling in. And if it hits the right pair, and the encoding for what RNA I'm on is in the color in that tail. So then in this case here, if I'm making a readout of 1,013 different things, I actually encode this information into 4 colors, 16 cycles, so 64 bits of information. So I cycle a reagent in. If there's one that has a landing pad for, say, the blue reporter group, and it lands and sticks, that means it's a hybridization event. That means that records as a blue that you see down here on your left-hand image. These are all single molecule images here. And you cycle that reagent off. The whole image goes dark, and you bring your new reporters in. They have a different little landing pad on the back there. And then in this particular case, I'm sort of saying that a little yellow reporter hit on the second cycle. So I would have measured down blue spots turning into yellow spots, and I'll blow up this image on the next slide so you can see it a little better. But you encode this information where all these little single molecule RNAs scattered over this entire tissue section are changing colors. And that color pattern identifies all the little transcripts that you're trying to measure. You can just encode, there's a sort of a schematic of the information. It's like a 4-bit, it's each bit has got 4 features associated with it, if you will. And you cycle through and you generate your full result image. So when you're looking at the raw data, that's sort of where it's coming from. If I blow those things up, you can see them a little better. Here's the little blue. There's a single RNA molecule there with one of our 20 nanometer sequencing barcodes on it. Here's another one. We cycle out to yellow, and there you can see a yellow one that's positive on this guy. We're cycling later on back to blue. They turn blue again, and you're doing this over your entire tissue section. That's how you encode it.
Can I have a quick question?
Yes, absolutely.
What happens when 2 RNA molecules are really close to each other?
Yep, it's an important thing. And so one of the key things is when you're doing imaging reagents, that's why we do no amplification on our imaging reagents, okay? So they're the smallest possible bright spots. So that's one reason that they're packed, roughly 30 dyes get packed into a 20 nanometer barcode. So making them small is good. That means the crowding happens later. You can crowd them in there a whole bunch, and I'll show you how many you can see at a time. But then the other way to control the crowding term is that I can control the series of sequences. So when I'm doing 4 colors at 16 cycles, some of these cycles are dark. So I am actually turning a whole bunch of things on and off. So you can actually have a couple of RNAs show up inside a single diffraction limited spot. But if it obeys a different on-off dark light cycle rule, you can actually resolve both of them, actually, even though they're in the same diffraction limited area, because I'm spreading the information out into different cycles. It doesn't mean eventually you will run into a crowded thing. It's pretty easy to see when you have multiple things that aren't even resolvable at that cycling, but you can actually do a lot. And it surprised me how much you can do. But anyway, it's a long-winded answer to your crowding question. Is that helpful?
Yeah, thanks.
Okay, so let's take a look at this thing and how it actually goes. I think kind of interesting to keep your tabs on how big is these data sets. So when I'm in there doing this cycling and doing these images over all these tissues here, and now you know these details of these little colors going on and off and so on and so forth. We generate, the raw data is about 10 terabytes that's coming out. That's a pretty good chunk of data. At 10 terabytes, actually luckily never actually has to move out of the SMI. Sits there. All these things then get decoded They get turned into actual digital genes or proteins. And all those digital genes and proteins have an X, Y, and Z coordinate that you can see over here. And once you've sort of digitized it at that level, you're down to about 1 terabyte of information, which is more manageable than 10. And you go one step farther, and then you actually go in and you start segmenting the cells on the right-hand side, where I actually know where one cell stops and another one starts. And now I know where all my RNAs are in these individual cells. Then you end up with a data set that's actually all the way down to about 1 terabyte. It actually has the gene identity, color codes, XYZ coordinates digitally now. The location of where that RNA is it in cell number or whatever, 1938 or 1205 or 304,003. They're all indexed that way, where they're at, cytoplasm. Your actual data set actually gets down to a pretty manageable thing that's down to about 1/10 of a tera. Okay, I see another one, a good question in there that I'll get to. 1/10 of a tera. But anyway, that's sort of the cycle of data through the system. Okay, this is sort of what it looks like now. I'm back into an FFPE melanoma sample here. You got the XYZ coordinates of everything. So I'm down into about a 1/10 of a tera database now with this study. And then you do exactly that thing I was talking about. You have all that information, you map it, you take the XYZ coordinates of everything, you reconstruct the tissue, and then you have all these extra genes so you can actually start looking at differential genes as I go through my mapping clusters. Like this is a differential heat map for a cluster. Let me find one that I can read with my glasses here. Let's take a look at cluster. I don't know. Yeah, cluster 5 is the first one in here. Oh, okay, cluster 5 right over there. This little green cluster of cells got a whole bunch of markers here that are hot for in that particular cell cluster, a UMAP cluster, if you will. And then you can just walk your way through the clusters, reading off the gene specific with those particular clusters. Now, let me tell you a little bit about the biology that comes out of here. This is our first panel we've been working with. It's a 1013 plex panel. And it was really designed to answer these real simple questions like, who am I? Where am I located? What am I doing in this location? What's my cell state and function? You can see roughly how many genes it takes to do those things. Cell typing and mapping actually only takes a couple of hundred genes. You can actually map a lot of cell types with that. Cell state and function, we devote about 270 genes for doing that. Cell-cell interaction, we're really interested on how the private lives of these cells as they talk to each other. So I really loaded up on all the cell-cell interaction and how the different receptor systems talk to each other and all of that. And then cells sometimes are affected by signals from far away, hormonal activities and things. So we wanted to have things like that. We sort of view this 1000-plex as sort of like table stakes, if you will. If you really want to look at a single cell, you want to understand these kinds of questions. It takes about 1,000 genes to do that. A half decent job of it. Okay, this is what it looks like when you're doing this, and we've got a lot of different types of FFPE samples run through the SMI at this stage. And I'm just showing you a handful of these. We do a lot of disease-based research here at Nanostring, so that's why most of these things look like different types of cancers and things: melanoma, non-small cell lung cancer, renal carcinoma, colorectal. We also look at things like tonsil, lymph nodes, those types of things. But in every case, you can do this high-plex, high-efficiency cell identification and then start looking at functional differences. I think, if you're a single cell person, one of the key things is that there's really no dropouts. And when you're doing this kind of work in a spatially resolved manner, it's not like single cells where you get these snippets of these high expressing genes and then you may pile 10,000 cells together to get a histogram. You are deep in every cell. There basically isn't any dark cells here. And you're measuring, you're going deep in the gene content of every single cell and no dropouts. Now, if you want to, you can actually put a bunch of tissues on a single SMI flow cell, and that really allows you to get more samples going at a time. This is one we were running the other day. Again, these are non-small cell lung cancer tissue arrays. What are we looking at? 1, 2, 3, 4, 5, 4, 8, 10, 12, 15 different tissue slices at a time, doing all this massive cell identification on all these particular types of tissues simultaneously. That definitely helps your throughput. Let me see, I'm gonna maybe answer a question or two here before I lunch, before I get too far behind them. This question on SMI technology, similar to SeqFish Plus and Long Kai's lab. One of the things that one of the major differences between us and a SeqFish Plus or a Merfish approach is that again, these are things we optimize for FFPE kinds of studies, where an individual RNA tends to be fragmented, so you don't have a lot of space. I can't tile a whole bunch of things along a large region because in an FFPE, those lesions aren't typically connected to each other. So we really designed it. We can actually go in there and do our high-plex profiling, and all I need is about 100 bases of contiguous RNA, and I can identify it. So that's one of the things, but it's similar. We encode, all these methods are going to encode information. So in that respect, the encoding, there's a million ways to encode information. We have our own way of encoding in these really small particles. But the other thing is that we look at a very small footprint of a little small footprint of the RNA that we are required to get. Okay. There's a question that's coming up on, okay, error recovery. That's right. When you encode 64 bits of information, give you a whole bunch of ways of encoding information. I'll show you what the false positives and the error rate looks like here in just a couple of slides. Okay, and I'll show you about proteins. So those are coming up. So the next couple of questions are already in my deck. So let me get back onto the flow of this. Okay, so now we're going into real FFP tissues. And you notice the importance of segmentation here. These cells aren't little balls or whatever. It's important. So we use the protein layer to determine the actual cell outlines. So we're using same kind of hybridization sequencing chemistry, but now we're on the back of an antibody. These antibodies are ganged up on membrane proteins. So that's how I generate those little aqua lines here. So I actually know where the RNAs go at the single cell level. And now this is really sort of a full-blown version of that. I view segmentation as still a slightly unsolved problem in this area. We spend a reasonable chunk of time on it, and I'm pretty happy to have gotten it to this place. So I'll just walk you through this. You can see the melanoma tissue over on the left-hand side. We're probing it with this 1000-plex. So inside that 1000-plex, but I really want to know where all those 1000-plex RNAs are in which particular cell. We also got a DAPI nuclear stain on there. So you can see now I'm going in there and generating them, blowing up the segmentation. So look at down in that lower middle. You can actually see cell outlines. You can see all their different shapes. We actually got the DAPI nucleus inside each one of those two, because these cells all don't line up like this. They're actually curving around, and it's all dependent on how the microtome chopped through this particular tissue. And that's where you can actually almost sense the planes of these cells where the microtome came through by looking at you're actually watching a nucleus rotate around in space a little bit, if you will. But, we're really happy to get it to this place. We're still going to be taking it much farther, but I really like being able to use this. Is a case where the protein layer of information really is super helpful into how to pack the RNA into the right cell, if you will. Okay. And there's a question about how to hit the membranes. I mean, I can't tell you all the membrane targets we hit there, but it's not exactly rocket science at that stage of the game. There's a whole bunch of integral membrane proteins that are pretty much common to almost all the tissues of the body. I think a variety of those. And are we done with our cocktail? Probably no. We'll continue to develop that. Okay, so this is what you get when start quantitating these things a little bit in terms of telling you how many RNAs are in each one of these cells. Here's a distribution over a number of cells here. You can see that sometimes we have cells where we're quantitating as many as 4,500 transcripts in a little tissue cell and maybe 10 microns on a side. So you can really, you can see a lot of transcripts in here. If you go back and look at the medium transcripts we're measuring, it's around 1,250 transcripts is our median per cell on these kind of tissues. And then on the right-hand side, I'm sort of quantitating them, the number of counts per cell. I'm using a couple of example case, high expressors, medium expressors, low expressors, and then we got these negative probability, negative probe 3, I think are labeled on here. And I'm going to start on a negative probes because it's something almost none of the high-plex RNA technology papers have in there. I don't know how they do it. They're absolutely essential, I think, for actually knowing what your real backgrounds are. So you guys probably in the RNA-seq business are familiar with these ERCC controls. These are sequences that don't exist inside human bodies on planet Earth anyway. So we actually, inside our assays, we always have these ERCC negative controls. I guess sequencing barcodes are going to combine to those things and recognize them and everything else. But it just turns out that those sets of sequences aren't in humans. It's really useful to do because then you get an idea of what your sort of real assay dependent backgrounds are. And then here they are, if I look at that thing. And if you actually quantitate these ERCC controls, the noise on this particular system is roughly 0.1 transcript per cell. Okay, so that gives you an idea. So when I look at something, that's my baseline background noise. If I look at a low expressor, say there's something that's 1 to 2 copies per cell, typically. This is some gene H4C3, whatever that thing is. But anyway, that's its distribution. It's known to be roughly 1 to 2 copies per cell. And you can see when you're quantitating that and your noise level is 1/10 of a transcript per cell, those low expressors are easy. You almost never miss them. That's why you don't have dark cells in these kinds of assays. They all, because you're seeing them at high efficiency, even at low counts. Then as you go to medium expressors and high expressors, you can sort of see the individual count layers on there. But I was pretty happy with this kind of background and being able to see the low expressors at this kind of level. Now you say to yourself that, again, like we did in genomics or whatever, we do all sorts of tricks to say, how do I know the things I'm measuring are right? Super fans, we made a ton of use of, there's a nice database of single cell, a nice deep RNA-seq database that we use a lot that was generated by the Broad. It's cancer cell line encyclopedia. It's about 1,000 cancer cell lines. They took like roughly 300,000 to 500,000 cells of those, do deep RNA-seq, and they made a database over those 1,000 cell lines. We take those exact same cell lines, we make FFPE pellets of those, and then we look at our 1,000-plex assays on them, and then we make plots where we can actually put the RNA-seq from CCLE on the x-axis, and then what I do, what I'm counting, in this case here, single cell counting, and it looks like we're adding up about 3,000 cells and just plotting those things out. And then how well do they correlate? And these are the kinds of correlations that we measure. These things map with the predicted RNA-seq and all these different cell lines. And those things are all different. If you ever look at those, there's solid tumors in there, liquid tumors in there. They are all very, very distinct cell lines. So when you get correlation of your 1,000-plex over all these cell lines, over all these different types of biology, you can be pretty darn convinced that the biology you're measuring in these assays actually represents the biology that are in these cells, and also the biology that are in the various tissue types that we're measuring. also. That's some of the kinds of validation we do. We also run RNA-seq on a lot of these things and quantitate the little RNA-seq dots with these higher-plex assay dots and all those types of things. So let me show you, let's do something with this thing. There's a couple of things. What's it like to hunt for rare T cells in something like here? We're in an FFPE kidney cancer case, and interacting with the investigator right a couple blocks away from me here, my labs here in Seattle at the Fred Hutch. He discovered a really fascinating class of T cells. I called them bystander T cells. And then there's bystanders, and then there's ones that are active, and we're on a hunt for the active ones. I won't go through the whole poster, obviously not enough time to do that. We showed a lot of this at AGBT first week of March. But this is the flow of information now on a real sample. There's an FFPE kidney sample. There's the UMAP clustering of all the cells we're monitoring in there. And you can see various classes, highlighted plasma, B cells, so on and so forth. And now remember, like I've said before, each one of those elements of the UMAP, each data point in a UMAP has got an XYZ coordinate associated with it. So then we can actually take that UMAP and then map it into space. And I sort of left it there so you can see the FOVs, the individual FOVs along the overall macrostructure of this kidney sample here in this upper right quadrant. So these are all ID'd now, probably about greater than 95% of the cells in that tissue would have been ID'd, identified from the RNA there. And we do things like in the lower left, we're actually looking at gene expression in what I call sort of neighborhoods. Like, are there groups of cells that tend to behave together in their gene expression pattern? And do these things spatially locate? And just like you do in genomics, DSP, you find out there's incredible action in terms of changes in gene expression pattern where boundaries meet each other, where one tissue boundary meets another. You see these all over the place in tissue. And the same thing happens here at the single cell level. You get regions where two different cell types meet along a boundary, and then you get unique gene expression that's linked uniquely to that boundary. And then sometimes there's gene expression patterns that span a wide spatial domain. So we combine these sort of functional neighborhoods with the cell identification, make overlap graphs on here. And then I'm just plotting specific types of T cells now in the right quadrant, right lower quadrant, and then superposing that on these sort of functional domains where weird things happen in tissues. And yeah, so you can explore these data sets. They're so information rich on so many aspects of biology, but just sort of giving you a feeling for sort of what first pass kind of things you can do. And this is the reason why all of us do spatial biology, because the information content is so dense and so rich. Okay, I want to give you another little bit of a flavor of a particular application of this, and I'm sort of excited about this one also. We're talking about CRISPR-Cas9 screening. So instead of just being passive with your tissue or passive with your cells, monitoring what they're doing, let's take and go proactive. Let's start engineering and do some engineering things like with CRISPR-Cas9. And then can we get functional readouts of cells at the same time we're engineering them? So this is what a CRISPR-Cas9 experiment looks like inside the Spatial Molecular Imager. What we're actually doing is using some of our barcodes, our sequencing barcodes, to actually in situ image the guide RNA, which guide RNA is in that particular cell at any given period of time. Sometimes you have multiple ones that will actually be in an individual cell, so that you can sort of see that over here on the right-hand side on a blow up. You take a particular cell, take that one with all the little green dots in the upper right-hand side, that would be just one guide RNA, and you can count how many are in there. And then you look right at the cell next to that green one, you see a mixture of both green and red in there. So at least probably two types of guide RNAs in that particular cell, no guide RNAs in the one cell over next to it, so on and so forth. So you end up with this map, spatial map of the guide RNAs and the combination guide RNAs that are in each individual cell. Now you got 1,000 plex worth of genes that are coming along for the ride here. So if you look at the guide RNAs, just look at the panel right underneath there, that'd be the 1,000 plex phenotypic readout of the change in those 1,000 genes that are related to the operations, the active operations doing at the guide RNA level. Okay, so in this particular investigator, this is actually a guy named Matt Friedman up at Dana-Farber in Boston, doing this work with Matt's lab. You know, he actually you know, he's a high throughput functional genomics person, and I'll tell you what he said about this experiment at the end, perhaps. He's interested in how antigen receptors work and his prostate cancer. So the cells you saw up there were called LNCAP cells, and he's knocking down, you can see various regulatory elements of the antigen receptor, really key receptor for prostate cancer and a number of other things. And then we're looking at the downstream effects of that in our functional assay. And then you can literally take assays like that and you read them off almost like, it's almost like a cartoon. That's more on the next slide. It's sort of like what it looks like, how many total transcripts. When you do these kinds of screens in here, what you see is that I got rich data on greater than 92% of all the cells that I'm measuring on. And if you ever do this thing at CRISPR-Cas9 at the single cell level, you know, nowhere near a 92% success rate on reading those cells. It's way, way down, as some of you perhaps know. Number of unique genes you're measuring per cell, and you know, another case where you get an incredibly rich set of information. Now with a readout like this, your controls are almost built in because you got enough plex, and I'm going to take you through one of these things because I think it's kind of interesting. I think we got enough time for this. Here's your guide RNAs. So let's take a look at slide number 5, where guide RNA number 5, and yeah, I don't know which one number 5, what the functional, what it's trying to do. Actually, I do know it knocks down some gene called KLK3, but anyway, so let's take a look at slide 5. That one's got guide RNAs in it. The one right next to it doesn't. And then I look at downstream signaling. Let's see how this guy reads out, actually. Let's maybe go. You can see in this particular case where we got, oh, okay, this case where it's a negative regulatory. Okay, so you knock something out up here, you actually turn on KLK3 or whatever. So you hit it here, it shows up positive here, you got tons of it. The cell right next door that didn't have the guide hit it, doesn't have any of it there. And just convince yourself that's not a dead cell number 1. It just doesn't have anything in it. You can read over here, the housekeeper associated with cell 1 is still there. So here's the housekeepers going. There's the lack of a knockout, it's not there. Here's the guide where it is. There's the functional effect of that guide. So you get all this high density information, and it's all self-contained in a single experiment. All the controls, the functional readouts, all that stuff is controlled. So it's really a high efficiency way of getting information out on these types of screens. I blew up one just so that it's not so confusing to see so many things. But here's the detected guides, no guides over here. Where the guides, then where there's no guides, you see a whole bunch of this particular target. Where there is, you don't see any. And you get these pairs and triplets of information across all the cells that you're measuring at super high efficiency.